Hello, Vienna, Austria. This is Dr. Donald Evenson, coming to you via recording from Brookings, South Dakota, USA. I wish I could have attended in person your European Society of Medicine's Congress, but since I could not, I trust that my message will come through via this recording. I am President and Director of SCSA Diagnostics. I'm also a Distinguished Professor, Emeritus, South Dakota State University. Also, I'm an adjunct professor at Sanford Medical School, Department OB/GYN, University of South Dakota. The title of my talk is Sperm DNA Fragmentation, or SDF, plays a significant role in couple infertility. Couple infertility is defined as attempting to conceive for at least 1 year without success. About 10 to 15% of women in ages 15 to 44 have difficulty becoming pregnant or staying pregnant. For eons of time, women have had a high level of blame for reproductive failure, with no pregnancy, miscarriages, no boy or child, genetic problems. For women, it is well known that her fertility clock is ticking. If you plot percent female likelihood of infertility in age groups, if you look at ages 35 to 39, it's already at 15%. Early 40s is 32%, late 40s is 60%, over 50 it's 100%. A long-time myth is that if a man has sperm and no pregnancy occurs to the woman in a reproductive age, it's the fault of the female patient. Message today is men. Your biological clock is ticking too. Current data show about 50% of couple infertility is due to the man, including no pregnancy, miscarriage, and birth defects. Question: could some infertility cases be due to the sperm not being able to penetrate the oocyte? In 1999, Dr. Palermo in Belgium invented a technique called HCID. HCID is an IVF mechanical injection of sperm into oocytes from fertile women. You have sperm going through a needle here into the oocyte for fertilization. It is thought this might help solve couple infertility. HCID did not solve the problem. We still obtain only 30 to 40% pregnancies per cycle. Suspected something was wrong with the sperm. My primary take-home question today is, will evaluation of sperm DNA fragmentation, or SDF, prove to be an efficient diagnostic tool in the prediction of pregnancy outcome? By definition, sperm DNA fragmentation is a single or double strand break in nuclear DNA with the potential loss of alteration of genetic information. Many agents break DNA. However, the primary agent is oxidative stress. In 1980, I was on faculty at the Sloan Kettering Research Institute in New York City. I was doing electron microscope studies on effects of cancer and chemotherapy on human sperm. I noted great heterogeneity of sperm nuclear chromatin structure. I asked the question, could abnormal sperm chromatin and DNA structure be related to infertility? I did a pilot experiment. I obtained sperm from two sources. First, sperm from proven fertile and subfertile bulls from the Eastern Artificial Insemination Cooperative. Also, sperm from recently proven fertile men and from men attending infertility clinics. I heated the sperm and buffer at 100 degrees for 5 minutes to partially open up DNA strand breaks at sites of broken DNA. Then I stained the sperm with acrid orange and measured the extent of DNA strand breaks. Arginine orange is a flat planar molecule of low molecular weight of 267. Arginine orange intercalates in a native double-stranded DNA and fluoresces green. Arginine orange stains on broken single-strand DNA and fluoresces red. Here's a fluorescent light micrograph of arginine-stained sperm. Your red sperm have fragmented DNA. Your green sperm with normal DNA staining green. However, it's almost impossible to quantitate the extent of red and green fluorescence in light microscopy. However, by light flow cytometry, you can pass these stained sperm into the glass flow channel of a flow cytometer. These sperm are then intersected by a laser beam. This excites the fluorescence that goes through a collecting lens, into a photomultiplier tube, and finally into a computer file. Here's an FCM cartoon cytogram of SSA and arginine-stained DNA. Here you have native DNA stainability versus green fluorescence on the y-axis, fragmented DNA or red fluorescence on the x-axis. Here are the normal green staining sperm. These DNA molecules, which are broken, are stained with via red fluorescence. I did a pioneering study. I found that subfertile bull sperm had 4 times more red fluorescence than sperm from fertile bulls. For men, infertility clinic patients had twice as much red fluorescence than sperm from fertile men. We published this study in the journal Science. The title was Relation of Mammalian Sperm Chromatin Heterogeneity to Fertility. Here's the fluorescence light microscope photograph. Here's the flow cytometry done for the first time. Here's green fluorescence versus red fluorescence. There are 5,000 dots here. Each dot represents a single sperm. Here's the sperm with normal unbroken DNA. These are the sperm with broken DNA with increased red fluorescence. From this, we developed the SCSA test. S stands for sperm, animal or human. C is for chromatin, a DNA complex with protein. S is for structure. There's a molecular structure of chromatin. A is for assay, which is the measurement of above. The SCSA protocol is quite simple. We have a diluted semen sample with sperm concentration of 1 to 2 million cells per mil. We do a 30-second pH 1.2 buffer treatment. This provides limited denaturation of DNA strand breaks at single sites of strand breaks. We stain with arginine orange. We measure 5,000 sperm by flow cytometry in less than 1 minute. Then we did a proof of concept of SCSA test in animal fertility studies, which are not possible with human studies. These were done with bulls, boars, and stallions. Here we have bull fertility rating versus percent DNA fragmentation index, which is the percent of sperm and ejaculate with measured sperm DNA strand breaks. We had semen from 58 bulls, and over the years it had 2 million inseminations. We plot here fertility rating of these bulls versus the percent of DFI. Those bulls with high fertility rating had low percent DFI, low fertility rating, higher percent DFI. We looked at boar fertility versus SCSA values. With 18 boars that were bred to 1,867 females. Here's percent DFI versus the fertility rate, value 0.6. Percent DFI versus litter number, 0.59. Interestingly, in this case, you have oocytes that have been fertilized with the sperm with broken DNA. These embryos are implanted in the uterus, and if the genes were broken for those that were critical for embryo development, the embryos died in utero. Therefore, the smaller litter size. We looked at stallions with 86 fertile stallions. Mean DFI was 16%. Subfertile stallions, we had 34. Mean DFI, 32%. We did some heterospermic insemination trials. This provides excellent and unique means to determine fertility outcomes versus DFI using different male phenotypes. Here's an example. You mix equal number of motile sperm from a black bull and a white bull. Inseminate 100 cows. If 80 black calves and 20 white calves are born, the sperm from the black bull are superior. Hypothesis: the black bull has better DNA integrity. Here we see SCSA data versus heterospermic bull competitive index with 9 phenotypically different bulls. You plot competitive index versus percent DFI. You see that high competitive index, you have low DFI. If you look at standard deviation of DFI, high competitive index, a low standard deviation of DFI. These studies were presented in a review paper I wrote in 2016 entitled The Sperm Chromatin Structure Assay and Other Sperm DNA Fragmentation Tests for Evaluation of Sperm Nuclear DNA Integrity as Related to Fertility. This is published in Animal Reproductive Science, Volume 169. After proving these SSA relative to animal fertility, we moved on to human pregnancy. In 1999, we worked with Dr. Zinnemann at Georgetown University Reproductive Medicine. We did 150 couples without infertility issues. We had timed intercourse at ovulation. We followed for 12 menstrual cycles. SCSA samples were taken every month until pregnancy or not. Here's an example of our data. Here's a couple that became pregnant in a manner of 7% DFI. We're plotting native DNA stability versus fragmented DNA. Here's the normal sperm without broken DNA. Here's a 7% of sperm with broken DNA and a high red fluorescence. In sharp contrast, here's a couple that did not become pregnant over the 12 months study. Again, here's the sperm without broken DNA. Here's the 65% of sperm with broken DNA. We published this paper in Human Reproduction. The title was Utility of Sperm Chromatin Structure as a Diagnostic and Prognostic Tool in the Human Fertility Clinic. This was published in 1999. This is now a highly quoted paper. In this study, those couples that became pregnant within the first 3 months of the study, there were 73. And the mean alpha T, which is directly related to percent DFI, had a value of 234. 40 couples became pregnant during months 4 through 12 at a value of 255. 31 couples did not achieve a pregnancy over 12 months with a value of 270. These are high statistical significance at P0 less than 1 and less than 0.001. From this study, and from other studies in other clinics around the world doing SCSA testing of clinical samples, we plotted here odds for live birth from high to low versus percent DFI. For a man with a 0 to 15% DFI, you have high odds for live birth. By 20%, this begins to fall off. By 25% DFI, this is the clinical threshold in which in vivo and IUI fertilization should be switched over to IVF-ICSI. One still obtains pregnancies at this point, but you have increased miscarriages. There are now 4 sperm DNA fragmentation tests. Our first in 1980, followed by Comet in 1988, Tunnel in 1993, SCD in 2003. All tests measure sperm DNA chromatin integrity as related to pregnancy outcomes. All are now used worldwide. A comparison of lab procedures for SDF tests show that SCSA and tunnel tests are done by flow cytometry, where you have 5,000 individual sperm per sample. In the Comet test, this is gel electrophoresis of broken DNA analyzed by light microscopy, 100 sperm per sample. The SCD test done by light microscopy, 500 sperm per sample. The Comet, SCD, and tunnel tests require many lab procedures and take hours of time. In sharp contrast, the SCSA tests can be done in less than 10 minutes. How useful are these sperm DNA fragmentation tests for the human infertility clinic? Dr. Blymo in 2017 published an article in which it was stated, many assays have been introduced into male infertility testing over the past 2 decades regarding varying aspects of spermatozoal competence. However, none have provided more clinical relevant data and insight into male fertility potential than the study of DNA fragmentation in the male gamete. Many conditions have been associated with elevated SDF levels, including varicocele, chronic illness, advanced paternal age, lifestyle, obesity, occupational environmental issues, medications, radiation, heat exposure. These conditions can promote sperm DNA fragmentation mainly by causing defective spermatogenesis, evoking abortive apoptosis, or increasing the generation of reactive oxygen species, or called ROS. Excessive ROS represents a significant causative factor of SDF in live sperm. Thresholds of 20 to 30% evaluated by SDF tests on neat semen are clinically useful for classifying infertile couples in a statistical probability of a longer time to achieve natural pregnancy and decreased chances of pregnancy by IUI, IVF, and ICSI, and increased miscarriage risk. Approximately 10 to 30% of couples with infertility have no apparent clinical or laboratory alterations in using conventional diagnostic approaches to explain their condition. Along these lines, a routine semen analysis is also unable to identify sperm defects at the molecular level. In a comparison of routine semen analysis versus SDF measures, we find that SDF rates in consecutive ejaculates are remarkably lower, about 9%, versus conventional semen parameters that range from 28% to 43% in count, motility, and morphology. About 40 to 50% of men with idiopathic infertility have elevated SDF rates. A decrease in SDF may allow these couples to achieve natural conception or eventually optimize the reproductive outcome of assisted reproductive treatments. A systematic review and meta-analysis of 28 studies and using SCSA, tunnel, and HCID tests indicated that a threshold of 20% DFI best discriminate fertile from infertile men, with an area under the curve of 0.84, sensitivity of 79%, specificity 86%. The oocyte repair machinery modulates the adverse effects of elevated SDF on embryo development and pregnancy. However, oocytes of advanced age women are less efficient in repairing sperm DNA damage. The persistence of DNA strand breaks and mutagenic bases might ultimately increase the risk of embryogenetic and epigenetic defects. We ask the question, how old is too old for fertility? This past year, we published a paper entitled Relationships Between Age of 25,445 Men Attending Infertility Clinics and Sperm DNA and Sperm Chromatin Structure Assay Defined Sperm DNA and Chromatin Integrity. We published it in Fertility and Sterility, Volume 114. In this paper, we have a figure where we plotted percent DFI versus age of men from 20 to 80. The red horizontal line is at the clinical threshold of 25% DFI. The blue line is a smooth curve. Note that from ages 20 to 40, you have one rate of increase of DFI. This rate doubles from 40 to 80. Note here, for example, that men from ages 20 to 25, almost all of the data points are below this threshold line. However, above this 25% threshold line, from 25 up to 80 is a significant increase. Of interest, note here that just a handful of men in the late 60s and 70s have a very low DFI for reasons unknown. Perhaps these men were drinking water at the fountain of youth. Here's a common clinical scenario. A wife of a couple struggling with infertility goes to the OB/GYN physician. Female examination is okay. Husband sent to urologist for semen analysis, he's okay. The doc asks, "Sir, have you fathered any kids?" He has, "Yes, 2 kids by previous marriage." The doc, who's not up to date on research, says, "Oh, you're fine. The problem is likely with your wife." No. In the same paper, we have a figure. We show the probability of reaching the 25% DFI level, critical threshold. Note that at ages, say at age 40, there's a 20% chance of reaching this clinical threshold. By age 50, it's a 40% chance. Age 60, there's a 60% chance. What are potential effects of having high SDF? It be stated that the health of infants could be impacted when natural or assisted inseminations are carried out with specimens with elevated SDF. The observed increase in mutational load in children of advanced age fathers is an example of the link between advanced paternal age, oxidative sperm DNA damage, and a range of pathologies. These pathologies include dominant genetic diseases in the offspring, achondroplasia, and neurodevelopment disorders such as autism, bipolar disease, and schizophrenia. The 2020 European Association of Urology guidelines on male infertility recommend SDF testing in couples who repeated pregnancy loss following natural conception IUI, and IVF ICSI. Also, men with unexplained infertility. Conclusion: SDF testing is well proven to provide clinical evidence to guide infertile couples on best procedures to achieve pregnancy or to use donor sperm,